Justin's just gonna watch him.
Are you just watching, Justin?
Yeah. He wants to make an entrance, like a true showman.
Are we live?
Hello, interwebs.
On the interwebs. Hello. Welcome. Let me see if this is working. Processing archive. Let's check back later. It says that it's not. Hello.
What? I have a lull contingent.
A lull contingent.
Whoop whoop. My neck's gonna hurt after this.
Yeah. Interesting. So this says the stream should be okay. We're going.
Oh, look, it's me on the internet.
But when we go to our channel, it's not
He's gonna make a grand entrance. I think he's gonna have to dance on stage when the show starts.
Hi guys! Hello, thanks for coming! Hello! Hello! You came, Justin. Fabulous.
I'm just hanging out in the chat room.
Oh yeah,
Yeah, I saw that.
I hang out in the chat room always. Says the stream health is good.
2:30 in the morning.
But then when I go to the YouTube channel, it does this.
They can see it though. So.
Yeah.
But maybe not.
My face has.
Maybe not use the bandwidth to try to watch it while we're.
I'm on a different Wi-Fi.
You
but
are?
Yes.
Oh.
Yeah, I will not use the Wi-Fi to watch it. I'm gonna close that up.
See what's on twis.org/live.
This is also an advertisement for the San Francisco Zoo.
Not condoned.
Everyone should go to the zoo. Not condoned advertisement.
No,
not
the
allowed.
views presented by TWIS are not that of the San Francisco Zoo or its affiliates. We get in so much trouble.
You know, it feels like it would be. It might be easier.
I've got a squishy bark.
Let me try. Let's try it.
thing where you like put a mirror in a mirror and it goes to infinity? What's that
Oh yeah, standing up. This is what I do at home. A mirror, an infinity mirror.
Yeah.
Forever and ever.
Do you really do that?
See, it's working.
I stand up. I do the show.
See, because I was wondering if I put this back there, would we like create a black hole? Because that would.
Very likely.
2:30 in the morning.
The standing up thing could work.
2:30 in the morning.
I like standing up.
I know,
when I
I'm
do
kind
the
of
show.
tempted to do that too.
You're the only one that isn't.
You look comfortable though.
Whoa.
You don't have to stand up.
I know, but I
You
usually
don't need
stand
to do
through the
this.
show too.
Alright.
But I am tangled.
Alright, we're 11 minutes in. Should we start a show?
Well, basically. Basically, it appears to be a device that converts an individual into digital information. What better way than to spend a Saturday night? 2:30 in the morning. What better way than to spend a Saturday night? 2:30 in the morning. 2:30 in the morning. I had to go for it and pray the gods were smiling on me.
Rock and roll?
Digital information. Digital information. What better way than to spend a Saturday night? Digital information. Digital information. What better way than to spend a Saturday night? In the morning, I had to go for it and pray the gods were smiling on me. What better ways than to spend a Saturday night?
No, I don't have a way to mute the mic.
Nope.
So my frequent coughing is going to sound a little more pronounced to me.
We're ready to go.
Is this our intro music?
This is not our intro
I
music.
haven't heard this before. We're going to be talking country science.
This is TWIS. This Week in Science, episode number 548, recorded on Thursday, January 7th, 2016. TWIS, live from STEMFest 2016. I'm Dr. Kiki, and today we are going to fill your heads with limpet love, free will, and jazz. But first,
Disclaimer, disclaimer, disclaimer. The following hour of programming is brought to you by the good people of science, reminding you that science is awesome. And by the highest IQs in the room fund, smart people supporting education outreach because we're smarter than everybody and that gets boring after a while. And by the men and women with curious minds in society who have no idea what the show is about but can't wait to find out. And special mention also goes to mass media, who have so passionately failed to cover science news that they have given us a brilliant niche. And last but not least, thanks to you, the listening audience. Without you, there truly wouldn't be any This Week in Science coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Science to Kiki and Blair.
good science to you, Justin, Blair, and everyone out there. I see you.
I can't see anybody.
I know, it's actually
I'm
dark.
completely blinded.
Welcome.
know you're there.
We know you're there. Welcome, everyone, to this week's episode of This Week in Science. And today we are broadcasting from the Crane Theater in Manhattan. We have a live audience. Yay! Let's hear you. Thank you so much for joining us here tonight. This is very exciting for us. I mean, Justin usually has bleachers at home.
I have a live
but
audience. I have
yeah.
it at home. Yeah, in my garage. I pack them in there.
Pack them in, bring them in. That's right. So anyway, as usual, we have a great show ahead. On tonight's show, I brought science news about vaccines, epigenetics? No, genetics, and King Kong. What do you have, Justin?
I've got a whole bunch of stories about brains.
Cool. That's it?
That's it. All brain stories.
Awesome. Blair.
Well, I have the usual invertebrate sex. I also have some jumping cocoons and some cane toads.
Cane toads.
Yeah.
We don't like them.
No.
No. All right, so let's dive right into this. So last year, Science magazine exclaimed that CRISPR, the gene editing technique, it was the discovery, the technique, the technology of the year for 2015. So we think it's going to be so big that we are starting this week on our weekly CRISPR Watch. Every week we're going to find a story about CRISPR, because I'm sure there's going to be at least one, if not more, every week. And the story this week is pretty amazing. For the first time ever, in adult living animals, we have edited genes in order to make a disorder better. So in mice, researchers used a virus-based adenovirus to get CRISPR and its Cas9 partner into muscle cells in a mouse model of Duchenne's muscular dystrophy. They were able to get this Cas9 CRISPR system in there by injecting the virus system into the muscles directly, also into the bloodstream of these mice. The mice that received the treatment versus those that had not received the treatment, but all had the muscular dystrophy, had much stronger muscle form than the others. So they actually had an improvement in the tension that their muscles could create. So the malformed protein that their bodies had been forming that was causing muscle weakness and degeneration actually was not cured, but made much, much better.
Yeah, and what's really wild about this is it's done in an animal that's alive, that has a disease, and the treatment goes in with the CRISPR-Cas9 and creates a treatment. This is something that's traditionally used to knock out or turn on a gene before it's born, but to be doing this as like a gene therapy in a living being, that's a next level of treatment that's possible with this.
Yeah, a lot of people are, CRISPR, it's the evil technology that's we're going to be editing our genomes.
And it is, and we will.
Yeah.
This is next.
But this is a good disease to start on. I mean, this is a pretty gnarly disease. People usually die by age, 25 when they get this disease?
Yeah.
So this is a great thing to kind of set our sights on and start trying to use CRISPR for good.
Using CRISPR for good, and there are going to be many, many more examples of this. And the note that we always like to make, this is research in mice, so it's not yet in people. There are many, many steps and many, many years before we actually see this being used for people who have Duchenne's muscular dystrophy, but this is a step in the right direction.
And again,
so
the brilliant thing about the CRISPR-Cas9 is this is the editing system in genetics that's being used everywhere throughout science currently. I mean, this is the one that's really taken off. The inventor patented it and then turned the patent over to the UC system, to the region. So he turned it over for them to let other researchers use it. He didn't try to simply profit off of it. So
there will be profit. So much profit. This is what's going to happen. Anyway, moving on in medical science for this last week. One really interesting study that caught my eye this week, a problem we run into in the winter season especially, are colds, flus, people running around going, get your flu shot, right? And then a lot of people going, I don't want to get my flu shot. I'm going to catch a cold from the flu shot. I know if you're listening to this in the audio version, you can't see me closing my eyes and shaking my head, but it causes me pain when I think of these things. So researchers have been trying to figure out why some people report adverse events when they get vaccinated. So a group of UK researchers published in Nature Immunology this week a study where they looked at 178 healthy individuals. So these are not people with any disease markers of any kind. They're healthy individuals. They were given the 2009 H1N1 swine flu shot. This is one that had pandemic potential, the swine flu did. And then they told people, if you have any kind of adverse reaction, anything, if you feel anything at all, a headache, you got a little tired, tell us. Let us know.
Let us know.
Yes. Definitely let us know then, for sure.
Yes, and so 20% of the individuals that were given the flu shot reported adverse events. They went and looked at those 20 people compared to the 20% of people who reported adverse events compared to those that did not. And they found that those who reported adverse events seemed to have higher levels of proteins related to B cells which are cells of the immune system, which happen to, and these specifically happen to be related to autoimmune disease. They had higher levels of proteins before and after getting the flu shot than people who did not report any adverse events. So people going in before getting their shot, even though they're completely healthy, have some kind of a profile. They had already some kind of a marker that maybe gave them a predilection to have some kind of a negative pathological, not pathological terribly, but a negative response to the vaccine.
So potentially you could test somebody in advance to see whether or not they should get the vaccine.
Right. Or you could say, well, you have this kind of a profile that you'll probably get a little tired and feel a little flu-y, but isn't it worse than actually getting the flu? And so it might give us a lot more better communication tools to be able to work with people to get them vaccinated. And it's not just the flu that's going to be the flu vaccine that's going to be affected by understanding how people's or immunology. So understanding why the body is reacting to itself in a particular way, why some cancer treatments work on people and not on others. So this line of research has much broader implications, but it's really neat. Maybe someday you'll know before you go to the doctor whether or not you're going to get that headache or die.
Which, in the long run, hopefully more people will get vaccines if they know that going in, then they won't have that weird mass hysteria of, oh, the flu shot gave me the flu.
Exactly.
And hopefully we'll all benefit from this.
Education,
Yeah.
right?
Oh.
Education about the self. It's all good. All right, this is This Week in Science. Justin, what'd you bring for us today?
Okay, show of hands, who here in the audience has free will? Good number of hands. Looks like nearly unanimous, right? Okay. So by the number of hands, it looks like this is a very self-deterministic audience. Of course, there has been pretty recent research that says this is not true. That before you actually put your hand up into the air, there was an automatic response in your brain that could be read by an EEG machine that would tell us whether or not you were going to raise your hand. Before your conscious brain got to interact and think, do I have the free will? Yeah, I think I do. Yes, I'll go. Already your hand was going to go up, and it could be read by the scientists. So this was sort of making a good argument for a deterministic nature of the brain, which has some reinforcement in other recent research. They can look at brains and tell if you're going to be, it can choose with about 80 to 90% accuracy whether or not you are a conservative or a liberal based on how you respond in your brain. So this is a little bit troubling, right? If you consider yourself a free thinker, to think that no matter what I do, my brain's just on auto and it's just going to choose for me. So they.
But it's patterns that have been formed for years, and right.
Right. Yeah. Absolutely.
Experience and your unique patterns.
Yeah. But again, if you're not getting to choose to raise your
Well,
hand,
but it's your subconscious that's choosing. So my subconscious has free will, but my
but
conscious
you don't.
doesn't.
But it doesn't. Maybe it doesn't either.
Right? Yeah.
Yeah, maybe it doesn't either. So okay, so some researchers who really didn't like this idea looked very hard at it and tried to set up an experiment where basically a duel with the computer, where it's like whoever moves first kind of loses, right? And the idea is, well, I guess you need to move first without the computer reading it, or the computer needs to know that you're going to move and announce that you are. Basically what happened was, by watching the brain, they were able to wave signal that passes through your brain before you raise your hand. And what people were able to do was edit it. They would decide with the auto to raise their hand, then fight the urge, fool the computer. So it turns out our free will isn't so much in what we choose, it's what we edit out of our choices. That's really where we get to assert free will in the process of the brain.
Awesome. Free.
That's a head scratcher.
Isn't it? Right.
You're free to edit.
I am
It's your-
free to not do things, is what you're saying.
Yeah.
Yes.
Okay.
Yeah.
Yes.
All right.
You're free to not do things. Your brain is going to do things for you. Right. Awesome. All right. Well, if you just tuned in, this is This Week in Science. And you know what time it is. It's time for Blair's Animal Corner.
Creature, bright and small. Biped, millipede, no pet. House animals, she's your girl. Except for giant pandas and squirrels. There's animal.
All very excited to talk to you about some awesome animals tonight. To start with, cocoons. Cocoons are pretty interesting. We talked about them a few times in 2015 as well. We talked kind of about the soup that the caterpillar turns into before it turns into a butterfly, and how we kind of thought it was just this homogeneous goo, but it turns out there's actually quite a bit of order to it. Well,
Like we decided, like minestrone.
like minestrone.
Still very weird. Still very strange.
Yeah. Yeah, just like minestrone. So inside those cocoons, there's a lot of interesting stuff going on. And in a few species, cocoons can actually move around while these animals are going through their transformation. And one really common example of that is Mexican jumping beans, right? Those are a moth species inside of a plant.
You mean they're not just beans that jump around?
They can jump though. They do, and they jump pretty high.
No.
They're pretty,
'Cause science.
pretty cool.
Because science.
Yeah.
Exactly.
So looking at a specific kind of cocooned wasp, this is the wasp Bathyplectes anuris. They are a really important wasp because they actually parasitize a weevil, the alfalfa weevil, that is really destructive on agriculture. So these guys are something that we have used in the past as a biological pesticide. And these cocoons, these guys can actually twitch around inside the cocoon and jump around in order to find themselves in favorable conditions. And so I actually have a video here that I'm going to screen share with you guys of these wasps jumping around. So right here we can see they are jumping around pretty much exactly like a Mexican jumping bean. It's pretty similar. And these guys do it for very specific reasons. So they like it dark, they like it cold, and they like it moist. And so they are more likely to jump around if they are moving from a warm area into a cold area, or a bright area into a dark area, or a dry area into a moist area. And so these guys do that so that they can grow bigger and stronger. But they have this really intense trade-off because they actually don't grow as large if they spend a bunch of energy jumping around. So they have a higher survival rate from jumping, but they're sometimes less fit after they've jumped around. So they really have to kind of do this cost-benefit analysis while they are minestrone
what a
in their
wonderful
cocoon.
life form.
While they're jumping
The
minestrone.
less you exercise, the fitter you'll get. This is...
Yeah, so these guys are really have a really tough decision when they're jumping around in these cocoons, and they find a way to survive. So this is a really important study for us to know what favorable conditions are for these guys so that they can continue to parasitize those terrible weevils that we don't want any part of.
Right, so make life really good for these jumping moths.
Yes, exactly.
Parasitize the
Yes.
weevils.
Yeah, so
Happy agriculture.
dark,
Dark,
cold,
moist.
moist. Yes,
Remember.
that's what we learned today. Dark, cold,
The
moist.
triangle of truth.
Yes. So those are the jumping cocoons. I think it's a pretty interesting phenomenon that we still are learning so much about with these guys. The other really cool story that I brought today about researchers looking at an interesting interaction between species is in Australia. And Australia have these really terrible invasives, the cane toad. So the cane toad or the marine toad, they were brought over to eat, again, this was a biological pesticide. They introduced them to eat locusts that were preying on the cane sugar. And so that's why these cane toads were brought in. They were called that to save the cane sugar. Unfortunately,
Oops,
No-
we
no
messed
natural
up
predator.
again.
yeah.
No
so-
natural predator.
So people who listen to this show a fair amount know that I will go on and on about invasive species and what a terrible idea it is pretty much 100% of the time. So in this case, they had no natural predators, and toads, a lot of toads, but these guys in particular, have a parotid gland. They have a poison gland. And that's how they've learned to survive despite living in South America where there are predators everywhere. But see, when you've evolved in a specific area, there's this give and take, and there are predators that have figured out to eat these guys when they're still tadpoles, and that balances out the population.
And
But in
so Australia, no predators eating
no
tadpoles.
predators.
Oops.
Yeah,
Again.
there are so many of these toads. They're moving at an estimated 40 to 60 kilometers a year. There's about 200 million of them in Australia.
So, a little bit dark, but they were being used at what do you call it? The golfing ranges.
Oh no.
Yeah, I mean, this is the level that Australia was resorting to try to eliminate them to no avail.
No
Well,
avail.
researchers have found a new way to not eliminate the cane toad, but protect the predators that are trying to eat them. When the cane toads are fully grown, they can get up to 8 inches wide. That's a pretty big toad. So we have one right here. And they are so big, they have so much poison. Oh, there's a general from the crowd.
And they're
look how
big,
cute
too.
he
They're
is.
big.
That when a large predator, for example, monitor lizards, when they go to try to eat the cane toad, they drop dead.
Right. Yeah.
Or they get paralyzed is the other thing that'll happen. So these monitor lizards, they are declining by around 90% right now, just from cane toads. So what researchers have done is
They brought in another invasive species to attack the cane toad.
good guess.
Is that
No.
They've
I think
actually
that'd be the
started
right move.
feeding cane toads to the lizards. But they're doing it when they're still babies, the cane toads. So they're feeding the cane toads to these monitor lizards when they're small enough that they give the lizards a really bad upset stomach, or maybe they'll even toss their cookies. But they won't die. And so once or twice doing that, they know no cane toads for me. It's
Right.
like one too many times with the tequila. You're like, never again.
Yeah, or at that restaurant with the bad fish sauce. Don't do it again.
Yeah, and it's actually working really well. So this is a case of researchers training wild animals to benefit the population.
So
And-
they'll still attack them, but they won't try to eat them?
No, they avoid them
No,
completely.
they'll avoid them.
But then how do you
Complete
reduce the numbers?
Well, that's a
Wait,
separate
that's
issue.
that's a different. Okay, got you.
First, save the lizards.
First, number 1,
Save
That
the
was
lizards.
step
Save
one,
the
get rid of
lizards.
frogs.
number 2,
Get rid of frogs.
get rid of the toads.
Toads. Get rid of the toads. Different.
Yeah.
Completely different.
So that's feeding toads to lizards for science. There we go.
There we go.
My last animal corner story of the day is all about slipper limpets. Can you say that 5 times fast?
Lipper.
Snippets, because when you turn them upside down and they're no. So the assumption was that it was chemicals, but it turns out these guys actually have to be touching to respond to each other's cues and transition or not.
That's interesting because you'd think in the aquatic environment that there is some kind of chemical signature being given off by the large animal in the pod, in the group, and then that would be in the water surrounding them all.
Yeah,
But
exactly.
it's physical touch.
They have to be touching, which, considering a snail, you wouldn't think about it
With
as
the
having
shell
that
to shell.
that complex of a social structure, but they actually have to touch each other. So in these experiments, they had 2 males that were slightly different in size, and they kept them in small cups containing seawater. And in some situations, there was a mesh barrier, and in other situations, they were able to have direct contact. And they found that when they were able to have direct contact, the larger limpet turned into a female sooner, and the smaller limpet delayed turning into a female longer. Yeah. So limpet sex more complicated than we thought.
It always turns out that way.
Yeah.
Little slippery invertebrates.
I don't know that I had a preordained level of complexity of
Oh, you
limpet sex
didn't?
before. No, I didn't.
Wait, doesn't
But-
everyone though?
No, I don't. It's one of the few species I really haven't thought about having sex, but
Well, I have some literature for you, Justin. We can talk about it later.
I think I've been thinking about invertebrate sex for about, 4 years now?
Yeah, you guys, tonight is my 4th anniversary on TWIS.
4
Yeah.
years. And I say 4 more years.
yeah, absolutely.
There we go. All right, everybody. Is that it for the Blair's Animal
That's it.
Corner?
Yeah.
We are going to take a very, very short break for some words, and we'll be back in just a few moments. Stay tuned for more This Week in Science. So everyone out there in the audience, if you do not have your 2016 TWIS calendar yet,
Supplies are still available.
supplies are still available. I brought some, $20 each if you're interested at the end of the show. Calendars!
And I will be.
That's amazing. You already got one. I sent you one. So we have our calendars. Blair did all of the artwork for the calendars in 2016. We have already gotten started on the list for art for, 2015. We have gotten started on the list for art for 2016 for the calendars to come. And what did we decide? One of the art.
Ideas for art in 2016. Boobies, tits, and shags. You guys will understand that in a little bit. Yeah. They're all birds. They're all birds.
That'd be good. Additionally, TWIS also has merchandise available online. You can head to our Zazzle store. So if you go to twis.org and click on that Zazzle store link, you can find all sorts of hats, t-shirts, mugs, I don't know, Christmas ornaments. Get ready for next year. We've got all sorts of stuff. Help support TWIS and proclaim your love for the science that we put out there.
Also has interesting things like Square Root Day, World Day of Laboratory Animals. They're all notated.
Yes.
So you'll never
My
get
personal favorite,
Calendar.
Species Requiem Day. That's where you
There are
just kinda
many
think-
days. You need this calendar to know what's going on in the world. Seriously, many, many days. TWIS is also supported by listeners like you. Your donations pay for everything that we do. This trip to New York, for instance. Hosting bandwidth, any fun things that we try to do. We appreciate any amount that you are able to give. $2, if that's it. Fantastic. Thank you so much for those $2. We accept donations a couple of ways. We have PayPal buttons all over our website. Just head on over to twis.org. We also have a Patreon account. Our account is Patreon, patreon.com/thisweekinscience. Head on over there, whatever your preference for how to donate. We appreciate whatever you can give. If you can't afford a donation, consider giving us some of your friends. Tell your friends about TWIS, your friends, your families, your coworkers. Use your social network for spreading the word of science news. And if you listen to us on iTunes, maybe consider posting a review of TWIS and giving the thumbs up to recent reviews that were helpful to you. That would help us out a lot and be just an easy way to support us that doesn't involve any cash. We thank you for all of your support. We really couldn't do any of this without you.
And we're back with more This Week in Science.
Hey, Justin, what you got?
I have threats.
Threats. What?
Yes, such as starvation or an attack by enemies. This is turning on genes in carpenter ants that change their behavior in ways that help their colonies survive. This is according to a new study co-authored by NYU Langone researchers and published January 1st, 2016. This is pretty recent. It's going to be in the journal Science. So more interestingly, perhaps even than that, is the related molecular pathways that they use to create this flip in behavior in these ants is present in humans. What they did was they found a way to block histone action in this. We'll call it histone A and histone D in these ants. And these ants are identical. These ants, when they're born, they're all identical. They're all the same. But some protect the colony, and some go out and forage and look for food and find their way into your kitchen. And so what they figured out, though, is there's these two genes that if they activate or deactivate, they can actually flip the ant's behavior. So that the forager becomes a protector and the protector becomes a forager. So just simply by activating or deactivating with histones, they're able to accomplish this. So they're not quite saying this can extend yet to human behavior or human activity, but because the molecular pathways are the same, they're thinking there's a number of human mental disorders that they could impact with this, because this is like the other story where they're starting to use the CRISPR to do alterations in something that's already alive, not in creating a new life form. They may be able to adjust human behavior this way.
Yeah, so the interesting thing is we have our genetic code, which we can't really change. I mean, we now with CRISPR-Cas9, I guess we can kind of change it. But the histones are these proteins that are wrapped up with the DNA, and they're what are considered an epigenetic control. And so they modulate the structure, the quaternary or the overarching structure the DNA molecule, the chromosome itself, and determine how proteins get expressed. So whether or not, if there's a histone wrapped up somewhere, it might determine whether or not a certain protein gets expressed and something ends up happening inside the cell. And so in this particular case, by swapping them, changing them, changing behavior. I mean, it's an interesting
Right. So, and we've seen previously the connection between the same genes when somebody's a very highly active person who's able to quantify large amounts of data. You see this very much in like the Silicon Valley, for instance. You have people who are really good at dealing with very large data sets. There's also a higher instance of autism in the same area, and it's the same genes. This is if they can actually, this is taking that whole idea of editing your genome to the next generation and trying to play God and be the creator of the genome that comes from you, and eliminates all that and says, no, you don't have to guess. You wait for the natural result and then learn how to adjust it if something isn't working out.
So like my child isn't as studious as I had hoped.
Hopefully you don't get it down to that point, but you look to use it
to the
for
histone clinic.
Yeah.
obvious disorders that are causing, that
I
have
really
social
wanted a football
ramifications.
player.
I didn't actually. I wanted a kicker, a football player, but a kicker, just somebody because they can't hit them.
Right.
And then if they can kick 40 yards by like high school, they get a free education somewhere.
Good goals. It's good to
Yep.
have these lofty goals.
# life goals.
Yeah, that's right. So in continuing to think about the idea of how things get passed along and epigenetic transformation of the genome, there have been questions for years about how genetic information or epigenetic information gets passed on from parents to offspring. So especially with dads, how does the information from the sperm, how does it carry epigenetic information? The sperm is this tiny little packet of DNA. It's got its mitochondria, so it goes really fast, shoots down there, hits a target, delivers the payload, but it's a very small payload. Where does all the stuff come from to determine things like children's metabolism? So
This is right. This is something we've seen in the past, where if you look at the grandfather or the father, you can actually hybrid offspring. So we knew this connection was there, but no idea how this was taking place.
Because it's not just in the genes. It's
not in the genes.
It's something else, and we haven't really understood how this stuff gets passed along. So a very recent study, a couple of groups have determined that it's more than DNA, it's tRNA. So transfer RNA is hitching a ride on the sperm as sperm travel down the epididymis to leave the body.
I've got one of those.
Yes, you do.
You certainly do.
Yes. So there are these little vacuoles with RNA. Transfer RNA is normally used to transfer RNA that goes from DNA to the ribosomes to become proteins. It's a transfer RNA from one place to the other, protein, there you go. It's involved in protein expression. But here, it's involved in a completely different process and is possibly involved in all sorts of gene expression across the board and in modulation of how genes are expressed in offspring at the embryonic level. And so a couple of research teams found that one group actually found that the transfer RNA hitch a ride on the sperm, and another group discovered that if male mice are fed either a high-fat diet or a low-fat diet, the high-fat diet males.
The experiences of the father passed on to the generation, and really skipped a generation, really more affected the generation to follow, the grandkids. So we've been following that for a while, but to see an actual mechanism fill in the correlation, it's always beautiful.
So what strikes me about this that seems interesting is it's so short term, and it is so dependent on what the male's eating that means
environment. I mean, who knows what factors are being transferred?
But that means that if we are in a society, if a child is
Which we are.
conceived in an area where where there are specific dietary changes over seasons, then that potentially means that you can expect certain differences in a child's metabolism based on when they were conceived in the year.
So like these fall kids could have. So the horoscopes have been right this whole time.
Yep.
Oh my goodness.
You heard it here.
But not
folks.
not for the reason they think.
No, but still, planet sponsor. Okay.
And another cool
Explains
study.
why all my Gemini friends are a little.
Too much cauliflower.
Oh.
Another interesting study related to what offspring get from their parents. There's been a question of why older parents seem to have children. So people who live longer but have children later in life, they seem to have offspring that don't live as long as they do. And so there's a question of, okay, well, the parents had a long life. There should be longevity conferred on the offspring. What is going on here? Researchers looking at a long-lived seabird called the European shag published a study in a periodical called Functional Ecology.
The aforementioned shag.
The shag. They looked at the birds. And the offspring that they were having, and they found that telomere length was involved. What they found is the chicks of older parents didn't start off with short telomeres. Now, telomeres we think are involved with aging. People who live longer tend to have longer telomeres, shorter life. And so the chicks start out with very long telomeres, normal-sized telomeres.
Quick question.
Yes?
Where are telomeres?
They
Where
are
would I
the caps
look
on
for
the
one?
ends of your chromosomes. So in your cells, at the ends of your DNA.
They're like
It
those
cap
little plastic things on the end of your shoelaces.
Yeah, so the question is, when, as they're growing in the nest, they experience rapid telomere loss. So the question now is, when they leave the nest and they are living their adult lives as short telomered birds, did it happen because the parents were busy living longer and not parenting? Or was there, so they're not giving as much parental care? Or is it just that they're older parents, and having older parents means that the chicks are going to have greater stress?
So next thing we need to do is go there and steal some baby shags and switch them around, right? And put them in different nests. And then we
Right.
can really know if it has to do with the parenting technique or the genes that were given.
You go do that.
Yeah, that's the and then we'll have to do that in humans next for the human trial. That'll take
We
a little.
have given some grad student a wonderful new dissertation project. Go. Be free.
We need a bell too.
Go play
so
with
for whenever
shags.
somebody gets their PhD, we can.
I know. Ding, ding, ding. Play with shags. How about something else, Justin?
This is scientists from Imperial College London have identified for the first time 2 clusters of genes responsible for intelligence, human intelligence. It's M1 and M3. This so-called gene networks appear to influence cognitive function, which includes memory, attention, processing speed, and reasoning. Scientists discovered that these 2 networks, which each contain hundreds of genes, are likely to be under the control of a master regulator switch. They're now keen to identify these switches and explore whether it might be feasible to manipulate them.
So you're saying
All right, show of hands again. Let's
make
get some
people
audience
smarter?
pictures. Who would willingly manipulate their brain to become more intelligent under the memory, attention, processing, speed, and reasoning categories?
For science.
A lot less than free will. This is actually, it might be the exact opposite of the free will hand raising, I think. It's like, I don't care about free will, just make me smart, right? In a study published in the journal Nature Neuroscience, the international team of researchers analyzed thousands of genes expressed in the human brain, then combined these results with genetic information from healthy people who had undergone IQ tests, and from people with neurological disorders such as autism spectrum disorder and intellectual disability. They conducted various computational analyses, comparisons in order to identify the gene networks that were influencing the healthy human cognitive abilities. Remarkably, they found, well, maybe not remarkably, a lot of overlap. lot of overlap with the disorders and those that were in the very healthy, because that's something we knew from other studies. Maybe they didn't read. They don't listen to this show. We've talked about this.
People have done it before. They knew.
Yeah, so they used the large data set. But, and of course, if we can manipulate these genes, enhance or better support their healthy activation, we could possibly create real, true smart drugs, not just amphetamines that keep you up and therefore you have to keep doing your homework. Or smart gene therapies, which again, in somebody who's already got a cognitive disorder, could be attended to.
Yeah, well, something like the nootropic field, that's the smart drug. That's where people are playing with drugs for intelligence. And
What could go wrong?
What could go wrong?
Well, it's a little scary. Like, the more you learn about how
Hordes of test-taking college students.
closely related high intelligence and mental disorders are, I mean, it's a little scary that they would play with that. And so I'd be, with everybody who didn't, I'm going with the majority. With everybody who did not raise your hand, I think I'd be with you. Until at some point that there was some task I was unable to complete. Like, if somebody handed me a Rubik's Cube, I might go ahead and say, yes, I'm ready for the gene therapy. I need this intelligence.
I just pull the stickers off. The Rubik's Cube.
That's cheating.
I know, cheater.
It's not actually related to intelligence.
No. You know who didn't cheat on his diet? King Kong.
What did he eat? Because that was a very small island to support
blonde
a creature
actresses,
of that
right?
size.
He was
He
not.
didn't eat the actresses, though.
He
That
was
the
not a
thing.
meat eater.
Oh.
Turns out, researchers, lots of people studying Gigantopithecus, the giant ape of the Pleistocene era, have wondered what the ape ate. There's been relatively little
my goodness.
fossil evidence. This ape has been estimated to vary from about 1.8 to 3.
Huge.
Yeah. You look at the size of that thing. Yeah. Anyway, in between 200 and 500 kilograms, so a weighty animal. And there hasn't been a lot of fossil evidence to be able to back up any ideas about the lifestyle of the giant ape of lore until recently. So a bunch of teeth were found. Teeth and jawbone came out of China, and researchers did analysis. So they were looking at the ratios of carbon-12 to carbon-13, nitrogen, oxygen, strontium, various things to get an idea of where the ape lived and what it ate. And they And they determined that it was a vegetarian, a very strict vegetarian.
Like a
too.
gorilla.
Like a gorilla. Didn't eat bamboo like a panda.
Good.
Yes. There were some hypotheses that the great ape only ate bamboo. Wrong.
Well, we know that's not a winning plan.
Right, but it was a panda lover. Yes. But the ape lived in forested regions. It did not really go out into the savannas, but this was the possible death knell for King Kong because it didn't want to go out into the savannas in search of other kinds of vegetation. It was very strict in its habitat and what it could eat. And that change happened at the end of or during the Pleistocene era, and forested areas became more savanna-like. Lost a habitat, lost a King Kong. Meh. But there are other ideas that there are other obligate or vegetarians like the orange one. What's the orange one?
The orangutan?
The orangutan. Okay. Thank you.
That's why we have a zoologist on
oranga.
the payroll here.
You're talking about the
Zoologist?
orangas.
The orangutan
All right.
also lives in forested habitat, very similar to what Gigantopithecus lived in, but because they eat more slowly and a little bit, they don't need as much food possibly. They don't have as much size as the great apes. So it's very possible that even though they are experiencing some amount of habitat loss, that it is not going to affect them as greatly. As
Well,
it did the
we've
ape.
seen this with a lot of different mammal species where they got really big and then just toned it down a little bit before they figured it out, right?
Yeah.
I mean, if you think about even elephants, there were much bigger versions of those before. And this is like the toned down version, what we have today.
Yeah, it's kind of like what you hope guys at the gym will do. Tone it down.
Okay. I learned something new about Kiki today.
right. Do we have any more stories? Is it time for our quick ones?
Yeah, I have a couple really quick stories. So out of Israel, new research on a robot locust has come out. This new robot is 3D printed. It's made out of the same stuff as Legos. It's called ABS plastic. And they were modeled after a locust. And using that technology, looking and studying how locusts jump, they were able to create this robot. And I have a picture right here. You can see him right there. So he has his little legs in the back. And these guys can jump really far. They can jump about 4.5 feet out, and they can jump over 11 feet up.
Wow.
And these guys can be made really cheaply for only about $100. So they're hoping that they can. So well, we don't want to, because then we'll have to let loose the toads that are robots, and then that could create a problem.
That could be fun.
Yeah.
Watch out for Robo-Locust.
Yes, and then the other quick story that I have is actually, it's my favorite story of the week. It's about GI Fridays or Applebee's or someplace mixed drinks.
Giant menus.
Giant menus. Well, it also actually 500 appetizer or dessert. Decide how many drinks you're going to have, just as a general, the weight of your waiter.
Wait staff.
Okay, a couple of dozen people got that message. The rest of marketing throughout the throughout the industry, we're going to see it.
Very carefully.
Use bait that smells like people. Oh. Right? Yeah,
Like,
so
aren't they like attracted to human exhalation?
carbon dioxide is one thing, but there are other aspects to human body odor that some mosquito species are more or less attracted to. And
That explains it.
scientists presenting at the meeting, they reported that they found a species of orchid that emits contained in human body odor.
I don't know if
It can attract its pretty much sole pollinator, the tiger mosquito. So they looked at all these orchids that were in a bog, and it seemed as if the orchids were only being pollinated by very specific species, even though there was a great diversity of different orchid species. Only specific pollinators were going to each one, and they said, I wonder why. And so they bagged them.
How did they bag the orchid?
Bagged them.
Okay. All right. Good.
And they analyzed the odors that were wafting into the air from the orchid, and this one particular species, body odor. You can't really smell it. And this is a common orchid that we have in the United States, and you might have it in your home.
Okay, so
It's
that
just-
means you'd get rid of it, right? Because it'll attract mosquitoes.
But
Okay.
the other point is that to draw them away, or you could figure out how to use these compounds to attract mosquitoes to take the mosquitoes and go.
It's a trap.
It's a tarp. That's right. And then my final story, or I guess I have a couple more, but this could be the last one. The periodic table. We haven't talked about the periodic table. You guys, we've Russian years, and finally chemistry had element 113, anuntrium, 115, septium, and 1 are official elements in the periodic table. Official names for these elements have not yet been decided. These are just placeholder names, and there are solicitations underway for names. So if you have ideas for names, you can. I
is
don't
that
think
too
that's going to work.
nascent
Yeah,
enough?
but there is currently a there to change magic.
That doesn't sound very sciencey to me.
Quick story, they put jazz musicians in an fMRI machine and had them play on a tiny keyboard, which I'm not sure how they managed that. But they found that sad song reward match.
Happy where when you're expressed
Of me going into further and further detail of how messed up it is.
the blues is usually talking about what's on your mind and giving you trouble, and maybe
I don't know. This is
Well,
really
it sort of does make sense of like how sad songs do sort of feel kind of self-affirming
Cathartic,
in a way.
yeah.
Yeah, very cathartic and great at work, where something that was lending itself to more creative thought might.
I don't
not
know
be.
that Pharrell song.
You seriously don't work and like have.
Yeah, my little personal dance party. Indicated when they think sad thoughts, feeling pathway that is involved in depression.
They're not really depressed. They're having a great time. Just nobody else is invited.
Yeah, fig story.
Fake.
Okay,
Did you say fake?
Eats figs. Do you like
Less
figs?
than. Oh no, it's
some
right
fig
there.
in it.
It's
I have a very
it's
exciting
good mix.
picture for
It's a
everyone who
good
eats
mix.
figs.
You ready for this?
in your figs.
In wild figs, however, there's this amazing ecosystem at work. Wow, it boggles my mind. So there's this tiny fig wasp that runs around the wild figs and pollinates them. But at the same time, while it's pollinating the wild fig, it is also dropping off bacteria, yeast, and worms. That's awesome, right? And then they all live in the fig together. And it's ecosystem.
And then you eat one, and a city is formed within your intestines.
Well, so the interesting point of this story is that some researchers went out and they're like, let's take a look at some of these worms. And they originally thought that there were different species of roundworms in different species of wild figs, and that they thought they were actually seeing different species of roundworm.
Science. This week in science.
Hey.
This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 Science.
Thank you,
This Week in
everyone, for
Science.
coming.
Science. And it shouldn't be news that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy.
And I have calendars, and I have some stickers and buttons and patches
So
if people are
everybody
interested.
listen to everything we say, and if you use our method instead
Calendars
of
are the
rolling
only
a
things
die,
that cost
we may
anything.
rid the world of toxoplasma gondii. Aye. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Science.